morning, dear students. Today we are going to learn about the functions of Golgi complex. In the last video, what we learned, we understood about what is Golgi, who discovered and what is its organization, from where it formed, all those things we have learned in the former video, in the last video. In this section, we will see about its functions. Main functions are forming secretory granules and its release. So, as these granules, as the Golgi complex continuously secretes granules, it may lose a portion of its membrane. So, that is recycling of what? Secretory vesicle membrane components. We will see in detail in the coming part of this video. So recycling of the secretory vesicle membrane components. The third function is glycosylation of proteins and lipids. Likewise, whenever proteins are formed from endoplasmic reticulum with the help of ribosome, it needs to undergo some additional modification, post translational modifications. And one such type of modification is glycosylation, and it is happening inside the Golgi complex. And formation of acrosome of the sperm cells. In the sperm cell, there is a structure named acrosome. I will just mention you what is that structure and what is its function in the coming part of the video. Then next function is formation of cell wall in plants. Okay, these are the main functions of Golgi complex. So we are going to handle each function separately one by one. First is formation of secretory granules and their release. How it forms the secretory granules and how they are releasing, where those vesicles are going, what are its fates, all those things we are going to understand. So this is all about its arrangement and its root. To understand its root, that's why I took this much. This is the nucleus and this is the endoplasmic reticulum, and adjacent to that we can see our Golgi complex, and this is the plasma membrane. Okay. So inside the endoplasmic reticulum, because of translation, what is forming? Some proteins are forming, and after it form there, it get attached with some address, a signal or label, and that label tells to where this protein need to transport it. So and from this endoplasmic reticulum, it comes out after loaded inside some vesicles. So vesicles are forming from the membrane of endoplasmic reticulum and form a separate vesicle, and name of those type of vesicle is transitional elements. What the name of such type of vesicles? Transitional elements. They are in transition between endoplasmic reticulum and Golgi complex. And then this transitional elements, it move further and proceed to the cis Golgi and get inside its vesicle and forms a vesicle, and then join with part of the cis Golgi and move from one cistern to another cistern and finally reach the trans Golgi portion. And from trans Golgi portion again, it bulges out to give rise to some vesicles, and that vesicle, it is called secretory granules. This secretory granules, then it goes and fuses with the plasma membrane if this protein is destined to release to the extracellular space. So if in that condition only, this secretory granule fuses with the plasma membrane and release that protein to the outside, that extracellular space. Okay, otherwise what will happen? If that protein is destined to be get incorporated to some other components, other cell organelles or compartments inside the cell, then it will incorporated to such compartment, maybe lysosome, mitochondria, like we so many phase destinations are there. So that's about the formation of secretory granules and their release. I hope it's clear to you. And this Golgi complex, it is predominant, especially it is more in a special type of cell, and the name of that cell are called goblet cell. Then what is this goblet cells and where we can find these goblet cells? These goblet cells are special type of cells seen intercalated, means in the space between the epithelial cells that line all our system wherever mucus need to be secreted, wherever a mucus layer need to create, in all those areas between the epithelial cells this goblet cells present, and they are responsible to secrete the mucus. So inside the such type of goblet cells this Golgi complex are abundant, is more. Okay, so that's about the first prime function of the Golgi complex. It is a transporter. It's just like a our post office and postman. It is transporting according to the label attached to the protein that is forming in from the endoplasmic reticulum, and depends upon that label, that address. It is transporting. It is conveying that protein to its final destination. So that is the secretory formation of secretory granules and release. Next function, recycling of the secretory vesicle membrane components. Because as continually from the Golgi, vesicles are come and join with the plasma membrane, gradually Golgi complex may disappear. So need to stop that. Need to recycle the secretory vesicle membrane components. How it is happening? This is plasma membrane, and this is a secretory granule or secretory vesicle. It is we have just learned that is joining with the plasma membrane and releasing its content to outside, and by that time that joined vesicle secretory granule from the Golgi complex, it is becoming the part of the plasma membrane. So by that way that much portion of the membrane is losing from the Golgi complex. It need to be recycled back to the Golgi complex. How it is happening? So as part of endocytosis. Endocytosis, you know what is the process? It is the process by which the cell is engulfing the particles from the external, means from its external environment. So endocytosis happening, and because of endocytosis, a portion of the plasma membrane, it forms a vesicle, and by that way this vesicle as it join with the trans Golgi again can recycle the lost part of the Golgi complex. So that is the recycling of the secretory vesicle membrane. In one way it give its component membrane portion to plasma membrane. In other means, through endocytosis, it regain that lost part again back to form the part of the Golgi. So that is about the recycling of the secretory vesicle membrane components. Next, we are going to see about the formation of lysosomes and microbodies. I already told in the beginning of the section that the Golgi complex play an important role in forming lysosomes and microbodies. How? The Golgi forms number of enzyme containing vesicle, because we know that enzymes all are proteins, and that proteins are forming inside the endoplasmic reticulum because of translation, and from it conveying to Golgi complex. And this enzyme, if it is destined to become the part of lysosome, that enzyme carrying that vesicle, granular vesicle, it retains inside the cell itself. It retains inside that cell itself, and later can form lysosome. Lysosome as well as can be converted to microbodies. Depends upon the type of protein that is loaded inside that vesicle. So in that way, Golgi forming lysosomes and microbodies. So from the Golgi, if it got a protein that is destined to be part of lysosome, it is forming a vesicle, and that vesicle later develops to a lysosome. So microbodies also forming in this way, and microbodies are of two types, peroxisome and glyoxysomes. And this peroxisome, they contain oxidative oxidation reaction carrying out enzymes, and this glyoxysome contains enzymes. What it is doing, it convert fat to carbohydrate. This glyoxysome contain enzymes that will convert fat to carbohydrate. And one more point, it present only in plants. Glyoxysomes only present in plants. So that's in that way, Golgi complex give rise to lysosomes and microbodies. And one more thing, inside the lysosome, certain proteins also present, and that proteins glycosylation also done by the Golgi. So in that way also, it helps in the formation of the lysosome. This is glycosylation. Glycosylation, adding glucose unit to some substrate. So Golgi complex glycosylate means it adds glucose unit to both lipids and proteins. After adding glucose or carbohydrate moiety to lipids and proteins, the lipids will become converted to glycosphingolipids, and proteins will be converted to glycoproteins. Okay, so glycosylation is another function of the Golgi complex, and that glycosylation is done by an enzyme. What's the name of that enzyme? Glycosyl transferase. It is present inside the Golgi complex. In the Golgi complex membrane, this enzyme present. Glycosyl transferase. Easy to memorize. It is the enzyme capable to transfer glucose moieties. So it is called a glycosyl transferase. And from where this glycosyl transferase is taking that sugar unit from some substrate, need to take the sugar moiety and to add to some protein. There should be some substrate, a donor, and usually the donor be a nucleotide like uridyl diphosphate, or so many nucleotides are available. Usually UDP stands as the sugar donor. So that's about the glycosylation. Then acrosome. Acrosome. What do you mean by acrosome? Acrosome is forming during spermatogenesis. And what is acrosome means? It is a membrane bound structure where we can see at the anterior end of the sperm cell. Anterior end of the sperm cell. And it contains inside that acrosome present so many enzyme and important so many hydrolytic enzymes, and among the hydrolytic enzyme, important one is hyaluronidase, and this enzyme it's a loaded by the Golgi complex. So this is a picture of that sperm, and this is the acrosome. It is a single membrane bound structure. It contains enormous amount of enzymes, and the abundant enzyme is hyaluronidase. It can cleave the membrane of the egg. Can cleave the membrane of the egg during the time of fertilization. So it's very important. And we can see that from the Golgi, in the former slide, I explained how the secretory granules are forming. The secretory granules, as it form, if the proteins, if the contents is destined to form the acrosome, then they stay here and they join with each other and form a big unit membrane bound vesicle carrying enormous amount of hydrolytic enzyme. That structure is acrosome. And without that structure, the sperm cannot fuse with the egg. If it cannot fuse with the egg, then there is no use of forming this type of cell. It can't do anything with the fertilization. So acrosome is an important structure in sperm. So forming acrosome, the Golgi complex play a lead role. And with the growth of acrosome, as the acrosome gain proper size, that means it is taking all its membrane from Golgi complex. It is clear that it is simple logic that Golgi complex gradually disappears because it is taking its origin from the Golgi complex itself by unioning so many secretory granules. In cell wall formation in plants, what is the role of Golgi complex? Okay. Okay. In cell wall formation, mm, in the plant cell wall, there present a glycoprotein. What's the name of that glycoprotein? It is hemicellulose. And that hemicellulose glycoprotein, it undergoes glycosylation. Uh, Glycosylation inside the Golgi complex. And after glycosylation, Golgi complex itself is secreting this hemicellulose to form the cell wall of plant. And also the cell wall. Plant cell wall contains so many enzymes. All those enzymes are also secreted by the Golgi complex. In that way, it play an important role in the cell wall formation. Then, how these Golgi bodies are forming? In the beginning of the section itself, I not clearly, but in a vague way, I told you that Golgi complex is forming from where? From endoplasmic reticulum. Golgi is forming from endoplasmic reticulum, that is nucleus, endoplasmic reticulum, and from here the vesicles are forming. It is joining with the Golgi. So it is clear that this Golgi is gaining its membranous portion from the endoplasmic reticulum. So in that sense, it is proposed that Golgi bodies take its origin from the endoplasmic reticulum, and that concept is postulated by Eisner and Novikov in 1962. Okay, with this we finish Golgi complex. Thank you.
I'm an icon, man. And my girl about a 10. I should fuck her in the Benz.